This topic is about fibrous proteins, and these proteins are basically structural proteins. So in the previous topic where we discussed myoglobin and hemoglobin, those are globular folded proteins, and these are the ones we often think of in terms of proteins, in terms of metabolism and being enzymatically active and catalyzing chemical reactions, or in the case of myoglobin and hemoglobin, binding oxygen. However, most of the proteins that we have in our bodies are fibrous proteins in terms of structural proteins. And so I wanted to have a topic devoted just to the features of these proteins and kind of explain the unique characteristics of them and how they deviate from the globular type proteins and that exist in aqueous solution, and because of that, it gives rise to their strength in terms of being structural proteins, in terms of providing an organism tissues that are required. So the learning objectives are shown below, and so we're just going to do some basic examples here and some of the chemical and biological features of these proteins. So fibrous proteins are basically structural proteins, and we start sequencing these, we find that they have very limited sequence variation, and they have a lot of repeating units over and over and over again. So know the basic properties, that they're composed of long extended chains, contain a lot of hydrophobic amino acids. This will promote these polypeptide chains to interact with one another through hydrophobic interactions. As mentioned, they have limited sequence variation, and they're insoluble in water due to this hydrophobicity, but they're very strong in terms of tensile strength, very strong proteins when they have their fibrous form. Examples would be your alpha keratin that you see present in fingernails, hair, and skin. Intermediate filaments in cells for structure of nuclei in the cytoplasm exist as alpha keratin units as well to provide some rigidity to cells in the cytoskeleton. Beta keratin, we see these in birds and fish in terms of feathers and scales. Collagen would be your bone matrix, tendons, and skin. And fibrin would be with your spider silk, spider webs, silkworms, silk, things of that nature. So each has a different chemical or amino acid composition and structural differences as well. So we're going to cover that in this topic. So if you were to look at the amino acid compositions of some of these fibrous proteins, you see the following. So what I wanted to highlight was the compositions of the contributions of the values shown in red here. So for example, in keratin, you have a high concentration of serine, glutamate, glutamine, and cysteine present. With fibrin, with the silks, you have a lot of glycine, alanine, and serine. Look at those numbers there. Everything else is probably very small. It's mostly glycine, alanine, and serine. If we look at collagen, we have a lot of glycine and alanine there too, but also a lot of proline. Look at that. Wow. That's a huge number, 22%. We talked about how proline being a secondary amine, a cyclic side chain, it has some properties of being a residue that's usually not found in helical regions because it kind of breaks up a helix formation because of this cis-trans isomerization and the cyclic side chain. It kind of kinks a helix, so they're usually not found there. They're present in turns, and so how come this protein have 22% of them? It's interesting. So we'll talk a little bit about these roles of these residues. And then if you look at all proteins here, you can see some of the numbers that these things are extremely different than what we would consider in terms of more of your globular proteins that we would think in terms of enzymes or binding proteins or transport proteins. So let's look at the alpha keratin as our first example. And this would be in hair and wool and nails and claws of outer skin layer of animals. And so the amino acids are primarily alanine, valine, leucine, isoleucine, methionine, and threonine, very hydrophobic. All right. And so recall when we look at helix-helix interactions that usually they're amphipathic helices that have one face that are predominantly hydrophobic residues, and the other face are hydrophilic residues, and the hydrophobic faces will come together to form a helix-helix interaction, a coiled coil. If you recall that domain that we talked about in the topic on protein quaternary structure. All right, so there's this driving force here with hydrophobic residues that have helices come together through hydrophobic interactions. If we look at keratin fibers, it's basically these sequences are in a helix, okay, and they'll interact with each other through a coil-coil interaction. So you can kind of think of it almost braiding two strands together. And then you have association of these two coiled coils interact with another coiled coil to get this protofilament. And then higher order interactions of these protofilaments will form this more complex filament here, which consists of 4 right-handed twisted protofilaments. So there's 4 of these in here. And so if there's 4 of these, that's like 8 of these, and that's like 16 of those. So you can see how it's like a rope where it's very highly twisted small alpha helices put together to give this formation of this filament. In addition, these keratin alpha helical structures have residues that have a lot of cysteine in them. And so these cysteines can form disulfide bonds. And so the more disulfide bonds you can form between these helical elements in the filament, the harder that structure becomes, the more rigid it becomes. So if you want to make that filament even tougher, make more disulfide bonds. So the alpha keratin then will be able to have a much harder filament if there's more cysteines in its sequence. So if we look at the hardest alpha keratin that's known, rhinoceros horn, it has about 18% of the amino acids are cross-linked with disulfide bonds. And so there's a lot of cysteine there, and forming a lot of these cross-links to make something very hard. We could take advantage of this in terms of the salon industry, in terms of using it to make permanents for different fashions of hair design. So basically your initial state of whatever your hair is in is going to have a certain number of disulfide bonds, and so the more disulfide bonds you have, it depends on your hair sequences. And so if you have curly hair versus straight hair, that can vary a little bit with some of the disulfide bonding that's present. And so you could break it up and and form different disulfide bonds. And that's what's happening when you do a permanent. So you basically will have your hair treated with something like a DTT, which we talked about in terms of breaking disulfide bonds in proteins, but this would be breaking disulfide bonds in your hair. Same thing. It's a chemical, and usually the chemical has a sulfurous, foul smelling odor because it's a reducing agent. DTT is more expensive than like beta-mercaptoethanol, so a lot of the permanent solutions will have the cheaper chemical reagent in there, and so you'll have this smell of this reducing agent to break these disulfide bonds. Then you set up the hair in curlers or however you want to have it be set. Then you treat it with an oxidizing agent to allow disulfide bonds to reform, and then that's going to hold that particular curl for a certain period of time. But over time the disulfide bonds would break and reform in their natural state, so eventually the curl will go back to whatever it is. So if you had curly hair, want straight hair, to get rid of disulfide, it can go back, or you set up a curl by pouring an oxidizing agent in there to preserve the curl, to do a permanent. Over time, as you wash and things like that, it will break it up as well. So this process of the properties of keratin are utilized when we in the hair industry. Also, even when you wash your hair, you notice it gets longer. That's because there's some disulfide bonding going between the alpha helices, right? And so if you were to break up some of that disulfide bonding with water and soap, the hair gets longer, but when it dries away and the water's driven off, those disulfide bonds reform, and then your hair goes back to its original length before you washed it. So we're not breaking disulfide bonds there. You need something as a reducing agent to do that. And then if you let it go naturally, it'll reset its normal disulfide bonding pattern. But if you want something artificially induced, you can add a oxidizing agent when you have a curl set up in there to do something different. So this is the biochemical basis of that in terms of different types of hairdos. Let's look at another fibrous protein, collagen, and this is the most dominant component of the skin, bones, teeth, tendons, and cartilage. And in terms of mammals, most of us are composed of collagen, and it's synthesized by fibroblast cells. And if we look at these sequences here, it's typically, as we saw in the previous table, glycine and a lot of proline and some alanine in there. So it's typically a lot of XY 3 amino acid residue repeats over and over again, where X position, the one in the middle, is usually proline, or it could be where X is something else, and then Y is hydroxyproline, which is proline with a hydroxyl group on it. And I mentioned before that the amino acids in the amino acids topics, that section, that the amino acids can get modified post-translationally, and you can have different groups that convey different chemistry to that particular amino acid. Hydroxyproline was one of those, and now you see how it is related here in terms of the structure of collagen. And so even though proline and glycine, and we know those residues are typically not involved in helical regions, in alpha helices of folded proteins, yet this repeating unit over and over again causes this to form a helix, but it's a left-handed helix, okay, but it still forms a helical structure, and these left-handed helices will come together as 3 helical bundles, coils, and they are right-handed, and they can get quite long, and that's shown here. So you can see the ball and stick diagram here. There's the space filling model here, and if you break it down, you can see it's like braiding, right? It's like you're braiding these helices together to form this triple helix structure. The hydroxyproline has a role because this hydroxyl group adds a potential hydrogen bond donor and acceptor on the side chain that doesn't exist as well. So it's very important to have the hydroxyproline present in helping to stabilize the collagen structure. And to have hydroxyproline, it's not going to be inserted into the protein during translation, but this is a post-translational modification. So you have enzymes that are required to hydroxylize or hydroxylate proline. And so this is a post-translational modification. One of the important cofactors involved is, in the process of allowing this to occur requires ascorbic acid or vitamin C. We'll talk about cofactors in the topic under cofactors, how they are small molecules or sometimes could be almost proteins in a sense in terms of helping to assist enzymatic reactions to occur. Often they're derived from vitamins and are modified a little bit to facilitate certain chemical reactions that the amino acids themselves cannot facilitate in a protein themselves through the side chains of the amino acids. And so this cofactor helps to catalyze unique chemical reactions that could not be done otherwise. So ascorbic acid or vitamin C is important for this proline hydroxylation. And so if you're deficient in vitamin C, you'll end up with what's called scurvy. And so this was common to sailors back in the 15th century when they were on ships for a long period of time and they ran out of their supplies and they didn't have any fresh fruits anymore, and so they would end up getting this condition called scurvy. And so they would have to land on some island somewhere and get some citrus fruits in order to get some vitamin C. And so that's where the term limey comes from when you talk about sailors. Hey, limey. It's because of the fact that, you know, they would be wanting citrus fruits. Limes is a citrus fruit, so that's kind of how that name came about. So we'll talk more about cofactors in another topic. But again, that was needed for the enzymes that hydroxylate the proline residue. So when we look at the alignment of these helical regions of collagen and look down on top of that triple helix, you can see how they're packed together. The proline, the hydroxyprolines are on the outside, and again, those hydroxylated prolines help with hydrogen bonding capabilities that wouldn't be possible just with proline. And the glycines are small side chains of just a hydrogen, and so they're hydrophobic and they'll kind of have the center there packed together. And so that's kind of the arrangements in terms of how these three helices will react. And then, of course, the hydrogen bonds within the helix will be the amide hydrogens and the carbonyl oxygens to form that ability to form the helical regions. Another aspect to add stability between these long triple helical collagen chains is to have covalent crosslinks form between these triple helices. In the keratins, we saw the disulfide bonds helping to facilitate that cross-linking, covalent linking across the different helical regions. Here, we're looking at the modification between a lysine residue of one helix and a modified lysine residue called the allolysine residue of another one. So it's a lysine that has been oxidized, and this is an aldehyde functionality, so it's very prone to nucleophilic attack by nucleophiles. And so a lysine residue here is showing deprotonated would be a great nucleophile to attack. Remember, the stronger the base, the greater the nucleophilicity. based on looking at chemical reactions in organic chemistry applications. And so this would be a great nucleophile to attack this electrophile here to form a shift base and basically have this modified cross-linked helical region. So that will add strength and stability to the collagen through this cross-linking, okay? So I just wanted to emphasize that this also has a cross-linking ability, but it's through these alanine residues. One of them has to be oxidized. That will add rigidity. If you look at how these fibers are organized as well, they are staggered, and so they have these little different repeating units that are staggered, and when they come together, so again, this adds to the strength. It's just like when you're making a wall of bricks. The bricks are staggered, right? They're not just the same brick just stacked up on top of each other directly, but you lay one row down, and then you stagger the other row on top of it. So that's kind of the concept here. These staggered strands add stability to the collagen. So in terms of these proteins, you can see they're very rigid, very strong, and they actually persist for millions of years in terms of some of the remains of fossils that have been around. So this is some work I did with collaboration with Dr. Schweitzer, in terms of a well-known paleontologist, and she's here at NC State. And so several years ago, we were able to get a grant and had a postdoc hired, Elena, and this is one of the papers we wrote. It's more of a review paper looking at, indeed, in case there is the case where we are finding fossils that are under certain conditions are highly preserved and well kept, that haven't been oxidized or damaged or tampered in a way that what is preserved there are some of the protein components from these collagens and keratins. And so if they've died under the conditions in which they ended up being preserved are ideal enough, these protein components from collagen and keratin can last for millions of years. And so this is kind of what the grant was to look at, is to try to understand more of these processes of preservation. How are these things designed to be preserved? How can they be preserved longer? How do what kind of other proteins may be present under these particular fossil examples? So you can kind of understand more of protein engineering better. How can we make better protein therapeutics that would last longer as well? So it has an application component, but also just a fundamental curiosity interest of what can we measure from some of these preserved samples using advanced new method development of protein extraction and mass spectrometry analysis. So just wanted to mention that even though they're structural proteins, there's still a lot of interesting things that can be discovered from them, even in samples that are millions of years old. The last example I wanted to look at was in terms of silk. And so this would be the silk that you would see in spiders and caterpillars. And so it is a repeating unit of glycine, serine, glycine, alanine, repeated over and over again. This one, however, is not helical, but are beta strands stabilized by hydrogen bonds to make these very efficient beta sheet structures. So shown here is the beta strand, and remember, in the beta strands we have alternating R groups above or below that strand, strand configuration, right? And so the repeating units over and over again provide the following that the glycines, for example, will reside on one face of that strand, and your alanines and serines would be on the other face of that strand. And so when these strands get together, you can see how the interactions occur. You have glycine being on the top, your alanine on the bottom, and it will interface with the other strand that has the alanines on that face to form hydrophobic interactions, and your glycines would be facing the other way, so the glycines would interact, then the alanines. So now you're stacking these strands based on the side chains. Alanine to alanines, hydrophobic interactions; glycines, glycines, hydrophobic interactions. But you can see the packing is different. The spacing here is larger for alanine than it is for glycine. And so you start stacking these strands together to get these huge sheets. And they're also pleated, right? So they're going up and down as well. And so you have this formation of this fibrin based on this stacking of these strands. And these pleated beta sheets are these fibrin, silk fibrin. So in solution, you can have these beta strands start to form, and notice it's anti-parallel here, and they'll start stacking on one another, and you end up with these very highly strong and elastic sandwiches, stacks of sheets that are formed. And so the connection between these sheets will be through these loop regions that are very amorphous. There's no structure to them, and so you can vary the connecting loops and how big you have these sheets to make various types of silks. And so nature does that through the organisms like spiders and caterpillars, but there's some folks doing work trying to understand the utilization of these features in spider silks to create a variety of products. So like bulletproof vests can be made through the design of polymers like spider silk, following these biological silk structures to create a very effective bulletproof vest, but weighs a lot less than your conventional bulletproof vest, or making other products as well. So there's a lot to be learned from nature, and we always look at what nature does and figure it out in terms of the basic understanding of the biochemistry, and then trying to apply that in some application from an engineering perspective. And so that's kind of what we do in terms of trying to understand the fundamental aspects of living systems, learning from that, and maybe making man-made products that would allow applications and some other things that don't exist. But nature has figured it out quite elegantly, and we're learning from nature and trying to improve on it. And it's debatable whether we improve on it or cause other problems because of our inventions. So that's more of a philosophical debate than we want to take right now. So this kind of wraps up the section on these fibrous proteins, but the main point is that by using repeating units of a few amino acids, you can confer some very novel and unusual structures that give rise to some strong material that is used in an organism's development. And we'll see the same thing in some of the other biomolecules, especially with carbohydrates, where they can be structural if they have a lot of repeating units as well. And so we'll save that for another topic. But have a good idea based on the learning outcomes of these particular structural fibrous proteins. So you have an idea of their composition and where they're found.